Hello, hello, hello. Happy Saturday afternoon. How is everyone on this fine Saturday? Roberto, why are you getting more and more anxious? Why are you getting more and more anxious? Yes, Alexander Tyler is in Colorado as well. He's up in Fort Collins. John Linick is in Colorado as well. He's in Denver. Sunny and 75. Nice. Esther is great. Excellent. All right. So let's get into our endocrine system. This comes from chapter 5 of our biology review. So this is going to move pretty quickly. Those of you who have been in channel sessions know how these work. So we're going to be talking about the different types of hormone signaling today as we get into class. So we have 4 very specific learning goals that we need to achieve in this hour. These 4 learning goals are understanding the different types of pathways that we have for signaling within our body, looking at the 2 primary methods of hormone signaling, looking at different hormone classes. So we're going to be talking about peptide hormones, amino acid hormones, and we're also going to be talking about steroid. And then identifying our negative and positive feedback loops. So this is really looking at that first core skill that we need to deal with on the MCAT, and that's having that science knowledge and being able to function within understanding what we're doing. So our first learning goal, we're going to start off with different signaling pathways within the body. So even though this class is called the endocrine system, we're going to be talking about different types of signaling, okay, including autocrine, juxtacrine, paracrine, so that we really understand the different types before we jump into our specific of endocrine system. All right, so first thing with endocrine signaling, different types of signaling, is when we're looking at signaling, this is just looking at very long distances, okay? When we're talking about the endocrine system, we're going throughout the whole body. And this really is just a form of cell-to-cell communication. When we look at this, hormones are secreted directly into the bloodstream. What is the other type of typical signaling that we talk about? I'll give you a hint. Pancreas does the other type of signaling as well. So typically we talk about endocrine and nice, Crystal. Very, very well done. Exocrine system. So exocrine is going to go directly where it needs to be. For instance, the pancreas is going to secrete its exocrine contents into where? Where does that go? Yeah, into the ducts, directly into which part of our body? Yeah, the duodenum, that section of our small intestine, that first section. Good. So endocrine is directly into the bloodstream, so this goes throughout our entire body. Then we're traveling to those locations, and it causes different changes in cellular expression depending on what it is that we are talking about. And we're going to get into this a little bit later. But before we do this, we want to talk about about paracrine, autocrine, and juxtacrine system. So when we talk about the paracrine system, paracrine is cell-to-cell communication, but this is nearby cells. So these are cells that are not touching each other, but they're very close by. And this happens through all of our extracellular fluid. Big thing we need to remember here is para is Greek for side by side, okay, being near each other. So let's talk about how this could work. Let's say our stimulus here is an allergen, okay? So a cell notices this allergen, and it's a mast cell. This mast cell is then going to secrete histidine. And this histidine is going to go to nearby cells, which is what we're seeing here. And they're being told, hey, we need to have an inflammatory response because we have an allergen here. This is all about proximity of the cells. Okay, they need to be fairly close to each other. So this is a great example, is looking at and thinking about an allergen within our body, histamines, and then that inflammatory response. So paracrines right next to each other, or very close to each other. Now, juxtacrine. Juxtacrine is cell-to-cell communications, and these guys are in contact with each other, okay? So they're either in close contact or directly adjacent to each other. And this can happen in a couple of different ways. there's always some sort of contact. So that can be that they are directly abutting each other or directly adjacent to each other. Or there can be something like this, where I have this nice little bridge, and this bridge is some sort of glycoprotein. And what happens is one cell is going to signal another, and that signal is going to go to another receptor, and these guys can now talk to each other. So this is going to go to that protein receptor that's on an adjacent cell. So they're very, very close, okay, or they're directly touching each other. So juxtacrine, second type, and then autocrine. What would autocrine mean? If I say auto, what does that mean to you guys? Self. Good, Lorene. Good, Esther, self. So this is something that is going to be a cell-cell communication where I release that signal and, hey, I'm going to accept that signal as well. And this can be something like a hormone that binds within a membrane, okay? And what that does then is going to release some sort of extracellular receptor, something to those receptors. Now, this is to self. It is not autocrine if it's going to another cell, okay? It only can go to itself. And this is nice because just like us, sometimes cells need to talk to themselves. So we need to know the difference between paracrine, juxtacrine, and autocrine. With that said, I think you guys have a great chance of getting this question correct. So here's what we're going to do. We're going to do this question together first. Some of the questions later I'm going to have you do on your own. But I want to make sure that we are really using our assess, plan, execute, and answer. So a nerve cell secretes acetylcholine, which activates an adjacent spinal nerve, eventually leading to the motion of the foot. This is an example of. So I look at this and I see that I've got different signaling in my answer choices, but there's a key word in here that is going to help me know what this is. What is that key word that I'm seeing? Ah, Esther, Lorene, Crystal. Good. You guys are saying adjacent. So this is really showing a signal type question. And if it's adjacent, okay, let's say maybe a neurotransmitter being released. What type of signaling is this? It's an adjacent cell. Ah, be careful. Be careful, okay? This one is, there's some sort of space in there. Okay, adjacent means nearby. They didn't say it was abutting or it was touching. Does that make sense to everybody? So it's adjacent can be nearby. They have to actually tell us that these two cells are in direct contact with each other, or they have some sort of bridge between them to be juxtacrine. Yes, yes. So it's a paracrine, exactly. So yes, an adjacent is a neighbor down the street, or many of us live in apartment complexes. There are apartments that are a floor up and 3 or 4 apartments over that are adjacent to us, but they're not in direct contact with us. Yes, Esther, good point. Esther's got a wonderful point here, that all of our nerve cells are going to use paracrine signaling. Why? Because there's that gap, okay? There's that space in there that they have to cross to get to the effector cell. So that's a great point, Esther. All right, there's no touching. It's got to be paracrine. Does this make sense to everybody? So be very, very careful. Adjacent means nearby. Adjacent doesn't mean to be abutting up next to. Excellent. Yeah, the synapse is the spacing. Absolutely, Esther. Absolutely. So that's where we get into the presynaptic versus postsynaptic cell when we're talking about our nerves. Excellent, excellent point. All right, number 2. You guys are ready for this. Here's what we're going to do. Is, Liz, if you can set me a timer for, I think you guys can do this in about 45 seconds. So we're going to give you 45 seconds to answer this question. Remember, use your assess, plan, execute, and answer. So you have 45 seconds. Good luck. Alrighty, guys, let's go ahead and look at this question. And I'll tell you, if you guys have been in a session with me before, I don't display the answers because everyone has a tendency to rush toward the answer that has been answered the most, or they'll change their answer. But I will tell you that 83% of us got this question correct. So this makes us very, very happy. Let's look at what we've got. A cell releases an autocrine signal that promotes cell division and proliferation. After the cell has released this hormone, so what I'm seeing is I've got cell division and proliferation. If I'm taking a single cell and dividing it, this means that I'm making clones of that particular cell. So what we need to do is figure out, and it makes sense because all the answer choices say clones. When I look at this, there's going to be multiple clones of the cell that is releasing the hormone. How do I know it's multiple clones of the cell releasing the hormone? Well, it's autocrine. So I'm going to release it. I'm going to signal myself that, hey, I need to divide. So this leads me to answer choice C. So autocrine is the same cell. Since the same cell is being stimulated to divide, there will be multiple clones of that cell. Now, let's look at answer choice A, because this is a tricky one that a lot of us choose. A cell releases, or there will be multiple clones of the cells around the cell that released it. If we're looking at clones that are around that cell that released it, what type of signaling is that? So if I release something and I tell others around me to divide, what kind of signaling is that? Yeah, Esther, that is paracrine. Good, Molly. Good. So take that question mark away, Esther. Be bold. Tell us, well, if I'm doing something that's adjacent to me or close to me, then that means that it has to be paracrine. All right? So it's a very, very small difference, but it really is a distinct difference. What we're looking for is, if it's autocrine, I'm signaling myself to divide. So I'm going to make multiple copies of myself. All right, very well done. That's it for our first goal. Let's look at our second goal. Our second goal here is looking at differentiating hormones by class, okay? And this is going to be various characteristics and identifying the two primary methods of hormone signaling. So we talked about other methods, paracrine, autocrine, juxtacrine, but now we're going to talk about different types of hormones, how different types of hormones signal each other. Let's look at hormone interactions within cell membranes. So there are two types of these when we're looking at cell membranes. The first type is going to be hydrophilic, okay? And the second type is going to be hydrophobic. We all know what this means. Hydrophilic means I love water. I'm on the outer or the inner side of the membrane. Okay, so I'm either outside the cell or directly touching the cytoplasm inside. Whereas that hydrophobic is going to be in that phospholipid bilayer in between. Okay, so if I'm looking at a cell membrane, if I am hydrophilic, can I cross through this hydrophobic region? Can I get through that? Oh, no. Excellent, because there's nothing in there that's got water. So I can't cross through that. Now let's think about this. What types of hormones, of our major two types of hormones that we are thinking about, what types of hormones would not be able to cross through that hydrophobic center? Peptide hormones. Why? Because they're charged, they're water-loving. This explains why they have surface receptors, and then they affect something within the cell. So now, if I ask you, well, what could cross this hydrophobic core? What would you tell me? Yeah, steroid hormones, absolutely, because they're going to go into the cell. So we're really talking about that phospholipid bilayer and how that helps us. Remember, the phospholipid bilayer is there to determine what comes into and out of the cell. So this is also helping us to determine what kind of hormones come into and out of the cell. All right, let's look at peptide hormones. So peptide hormones are made up of amino acids. We know this. Now, be careful, this is not the same as an amino acid-derived hormone, okay, where it'll be just one amino acid. This is a group of amino acids. So this is a nice peptide chain. This is derived from larger precursor polypeptides, so it can be cleaved, but it's still, even though it's smaller, going to be multiple amino acids. Then I look at this and I know that this is going to bind to extracellular receptors because it can't pass through that hydrophobic core. This is not surprising to us. We all know that when we're looking at peptide hormones, they're going to not be able to cross that phospholipid bilayer. Now, this is important. They have a fast onset and they're short acting. So let's talk about why they have a fast onset. Once I bind, I'm going to change the shape of some sort of receptor inside there, and it causes a cascade, and I get a pathway. What happens at the end of that pathway? What are we going to bind to that is already present in my cytoplasm? What are we going to bind to that's already present in my cytoplasm, which is why this is so fast? Yes, Crystal, a protein that is already present. So if this protein is already present, then I'm either turning it on or I'm turning it off. So we can see this fast onset because the protein's already present. So we're looking for, is it no longer working or is it now working? So this is going to be some sort of protein within the cell. That's why it's fast acting. But why is it short acting? Why is it not long term? Explain to me why it's not long term when we're looking at this. Why is this not long term? See lots and lots of us typing. This makes us happy. Part of it is feedback. Part of it is negative feedback, definitely. But another part of it is, yeah, it makes those conformational changes in that protein, which is not permanent. So we're getting there. Those are not permanent. Also, if I make a protein, does it last forever within the cytoplasm of my cell? No, it degrades. So once that protein that has been acted upon degrades, I no longer see whatever that action is. Okay, so that's one of the reasons that it's very short acting, is those proteins degrade. They don't last forever. So very quick, very short acting. Here's an example. Oxytocin, which we all know what oxytocin does. So if we look at this, this is definitely a peptide because it's got multiple amino acids. So what does oxytocin do? Let's remind ourselves, this is definitely one of the proteins we need to know that's made by the hypothalamus and released by the posterior pituitary. Yep, uterine contractions, uterine contractions. And let's think about this for a second. What type of feedback system is this? Because you guys mentioned feedback. What type of feedback system is this? Ah, positive feedback. How many positive feedback systems do we have in our body? Lots or very few? Yeah, not many. Not many at all. Oxytocin is the one that we need to know. Okay, pretty much everything else is negative feedback that we need to know for the MCAT. But this is the positive feedback we need to know. Why are there so few positive feedback loops in our body? Why is oxytocin one of the few of them? Yeah, regulation. They are hard to control. They're hard to regulate. Exactly. So we don't want a lot of positive feedback systems in our body. This is the one that you absolutely need to know as a positive feedback. Okay? Now, let's look at our peptides here. So if I have a peptide, I have this intracellular protein. So what's going to happen here? My peptide hormone is going to come. It's going to bind to its receptor. Dun, dun, dun. Notice it bounced off the membrane. Why? Because, well, it can't go through the membrane, can't pass through that hydrophobic core. So it binds to its receptor, which then turns it on. Okay, we went from a red light to a green light. And we typically make something like CAMP. Okay, and we know that that is going to give us a signal downstream. So it upregulates a kinase. So I have this enzyme complex. Well, if I started with ATP and I have AMP, this is going to be a kinase, which means I'm moving a phosphate from one group to another. So I'm now going to phosphorylate this enzyme. And this phosphorylation can affect transcription. It can also affect a protein that is already there. Okay, so these are second messenger systems that really help to affect those signaling cascades. All right, now, there are lots and lots of peptide hormones. question is, how do we remember our peptide hormones? Okay, so what we're going to do is we're going to give ourselves a mnemonic. You guys ready for the mnemonic? It is insane, I-N. It's insane how frequently peptide hormones begin or end with I-N. Okay, it's insane how frequently peptide hormones begin or end with IN. So let's look at a couple. Insulin, oxytocin, prolactin, vasopressin, somatostatin. Okay, this is nice. We've got this. Are there exceptions? Of course, there are always exceptions. A couple of the exceptions that you need to know are going to be ACTH, adrenocorticotropic hormone, and glucagon. Okay, so here are two of the exceptions you need to know. But now, if I get a passage on test day where they give me a hormone that I've never seen before, and I see that it ends in IN or it begins in IN, I now know this is going to be a peptide hormone. Okay? If there's not an indication like that of what type of hormone it is, they will tell you. They will say whatever the name is, which is a peptide hormone. But now that you know this mnemonic, you now know when you see these that these are going to be peptide hormones. Gotta love it. Love our mnemonics. So let's now look at steroid hormones. Steroid hormones, slower onset, okay, takes a while to see the effect, but it's longer lasting. So tell us why it's a slower onset. Why don't I see this affecting my cell right away? Why is it a slower onset? It's got to go inside the cell. It's got to get through that hydrophobic core. Then if it can cross my plasma membrane, what else does it cross? There's another membrane it can cross. It crosses the nuclear membrane to, as Alexander says, regulate transcription. All right, so it binds directly to my DNA, regulates my transcription, turns a new gene on, turns an old gene off. So this is why it takes longer to see it. Now, because it's bound to my DNA, this is also a longer lasting change than our peptides. Sometimes this can actually be a permanent change. Give us examples of a couple of steroid hormones you know that can make permanent changes within the human body. Oh, estrogen, testosterone, absolutely, our sex hormones. Okay, here's one way that you can remember that these guys not only are long-lasting, but they can be permanent. Does everybody remember puberty? I know you don't want to. I just gave you some really nasty flashbacks potentially. Okay, but yeah, we all remember puberty. Okay, could you imagine if these were not permanent? Having to go backwards through puberty at some point in your life? Just imagine that for a second. Everyone's going, oh wait, it was bad enough the first time. What's going to happen going backwards? Okay, so this is how we can remember that sometimes these are permanent. Now, are they always permanent? No. Okay, let's think about cortisol. Okay, that's our glucose metabolism. That's not something that is always happening. Or aldosterone. Where does aldosterone act? And what does it do? It's another good steroid hormone. Yeah, it acts in my nephrons. It acts in the distal convoluted tubules and a little bit of the collecting duct, and it causes us to raise our blood pressure by reabsorbing sodium, and water is going to passively follow that. Do we want that to be permanent? Do we want to constantly be increasing the amount of sodium and water that we reabsorb in our kidneys? Oh, no, no. Yeah, Esther, then we would have high blood pressure all the time. So even though they can be longer lasting, because we have to wait till that blood pressure goes up, or we have to wait till that glucose metabolism is where it needs to be. A lot of them, most of them, are not permanent, but they are still very long lasting. Okay? Does that make sense to everyone, how they can be long lasting but not permanent? Excellent. Perfect. All right, so let's talk a little bit more about these. We know they're not water soluble because they're made from cholesterol, which means they are hydrophobic. Okay, they really, really just don't like that water, so they're not water soluble. This means that they have to be carried by proteins in the bloodstream. This is different than peptide hormones. Peptide hormones can be dissolved in our bloodstream and just be moving through our bloodstream. But these guys, there has to be a carrier protein, or they're just going to curl up in a little ball and they're not going to want to go anywhere. We need to get them through our entire bloodstream, through our entire body. So here's some examples of the carriers. A sex hormone can bind to a globulin, okay, or albumin, okay. Albumin is our major plasma protein. We should all know that one. That's a great way to carry some of the hormones in our body, okay. So they have to be carried by a globulin. They can be carried by albumin. So let's look at the structure of this. What do you notice about this hydrocarbon structure here? What do you notice about it? Oh yeah, lots and lots of aromaticity. We see this a lot in steroid hormones. You see a lot of aromatic chains. Yeah, and they're not polar, and they're big. But the big thing is with those aromatic chains, these guys are so hydrophobic that they've got to have some way to get them. Yeah, they've got huge hydrophobic chains. Absolutely. All right, so let's look at how this works. So before we saw the peptide hormone, it came in and it bounced right off of the plasma membrane. Well, this isn't going to bounce off. These guys are going to be able to go straight through. Notice they're binding to something in the nucleus, their receptor. And then that receptor hormone complex is going to go and it's going to bind to our DNA, either turning the transcription on of a new gene or turning a transcription off of an old gene. So this is changing our transcription, which you guys already said is going to happen. So this is going to go straight through. It's going to go through that nuclear membrane, which is what those two blue lines are. And that hormone receptor complex is going to bind directly to our DNA. Okay, so we know that. Now, we need to know how to remember our steroid hormones. We already know it's insane how many peptide hormones have IN either at the beginning or end of their name. We have another mnemonic for our steroid hormones. Okay, so let's look at our mnemonic. Many have, wait a minute, give me a name. You guys already said testosterone. Okay, progesterone, aldosterone. Do you guys already see a pattern there? Yeah, ONE. So many of them end in ONE, OL, OL, or OID. Okay, so testosterone, progesterone, aldosterone. Estradiol. Okay, estradiol is a form of estrogen. It's a preform of estrogen. Okay, glucocorticoids. So when I see ON, AL, or OID, that is a really, really good indication that this is a steroid hormone. Again, this lets me know in a passage if they give me a steroid hormone, then I don't necessarily know. I can see this ending and know, that's got to be a steroid hormone. Yes, and good, Esther, nice, I like that. Estradiol does share the same ending as cholesterol, which makes sense because what are steroid hormones made from? What is their precursor? Cholesterol, absolutely. So our mnemonic, Miriam, here is just many have the suffix, there's not a nice saying, sorry, I don't have a clever saying for this one. Many have the suffix of ON, AL, or OID. And OID makes sense because it's a steroid, okay? So cholesterol, AL has that same ending. Steroid, we've got that OID. And then we've got our ONE, which we know because we know testosterone, progesterone, aldosterone. Those are common ones we should know. Now, the weird exception here is estrogen, but we all know estrogen is one of our major steroid hormones in our bodies, especially in females. Okay, and we know that the precursor to that is estradiol. Okay, so we've got these. We can do these. Now, that's two types of hormones. Those are the two major types of hormones that we need to know off the top of our head. Our third type are less common, but still very, very important. And these are going to be our amino acid derivatives. Okay, again, they're less common, but they're still very, very important. When we look at these are things like epinephrine, norepinephrine, thyroxine, which is T4, and then T3. Okay, so we've got T4, we've got T3. When we look at those two, T4 and T3, remember what that 4 and the 3 are, is the number of iodines attached. Okay, T4 is actually released, and then T3 is the more active form that we get at our cells. Okay, as we look at this, we've seen these. Do you guys see an ending that looks really familiar or really common in our amino acid derivatives? Yep, INE. Okay, so let's look at a couple of different things. They're typically made from 1 or 2 amino acids. This makes them very different from peptide hormones, which are made from multiple amino acids. So they really are peptides. They're small peptides, but they really are peptides. Okay? Yes, Esther, good. Amino acids. Good. Now, here's the cool thing about amino acid-derived hormones. They can act like either a peptide hormone, which means that they're going to have an extracellular receptor, or they can act like a steroid hormone, which means they're going to have an intracellular receptor. And this is going to all be dependent upon their structure. Okay? So let's look at how this works. Here's tyrosine. And we absolutely do need to know our amino acids for test day. So we should know what this looks like. Excuse me. Sorry about that. It's spring in Colorado. What can I say? Okay. So when I look at tyrosine, here's thyroxine. Okay, and we know thyroxine is T4, and I can see 1, 2, 3, 4 iodines on here. So when I look at this, this is an amino acid derivative. Why? Because it's a derivative of our tyrosine. Now, how does the body transport a thyroid hormone? Look at this thyroid hormone. Is this more similar in structure to an amino acid, or is this more similar in structure to maybe a steroid hormone? Look at the shapes. Remember what testosterone looked like. Ah, this is more like a steroid. Why? Because we have these nice aromatic rings. And we know that there are only a few of our amino acids that have aromatic rings. So if it's got these aromatic rings, it looks more like a steroid. How is it going to be transported if we're talking about specifically a thyroid hormone? Ah, yes, a carrier protein. A carrier protein could be albumin, okay, could be another type of globulin, but it has to have that steroid protein. Ah, okay, couple of great questions. Yes, we have hydroxyl groups on here, and yes, we have iodine, but remember with iodine and polarity. Iodine can be polar, okay, it can cause polarity, but is this as electronegative as something like fluorine? Think about where it is on our periodic table. Is this going to be, yeah, not quite, not quite. So even though it has these iodines, remember we have this aromatic ring here, okay? So we're going to be constantly moving the electrons around in this aromatic ring. Very, very stable because of the aromaticity, okay? So it distributes that negative charge, absolutely. Absolutely, good. And remember testosterone. Testosterone, when I asked you guys to look at it, the first thing you said is, wow, look at all of those aromatic rings. Okay? We have 2 nice big aromatic rings here. The only difference between thyroxine, which is T4, and T3 is the removal of 1 iodine. Okay? So because we've got these aromatic rings, because it's really not polar, okay, we're going to be able to distribute that negative charge. This looks more like a steroid hormone. Okay? And Esther, it's true, it's big, it's bulky. Okay, not all amino acids are this big and bulky. Okay? So you guys have got some great points here and some great questions that we're being asked about. Okay? All right. So this one, if I look at this looks more like a steroid. I need some sort of carrier protein. All right, let's look at our summary. We've all seen charts like this before. We're really going to be talking about peptide and steroid hormones. Why? Because they're significantly more common and they're significantly more important. Okay? Amino acid-derived hormones, they're still important. We all have to know epinephrine, norepinephrine, T3, T4. Okay? But the more important and more common are going to be the peptide and the steroid. So when we look at this, precursor means, oh, what is it made from? Permeability means, can it cross that hydrophobic core of my cell membrane? Where is transportation? Where are the receptors? What is its mechanism of action? How long before I see the results? How long does it last? And then examples. And we've pretty much gone through this. So a peptide hormone is a precursor of amino acids, a longer peptide that gets cleaved. Okay? Whereas steroid, you guys all said, was cholesterol. Peptides are polar, they're not permeable. Steroids are nonpolar, hydrophobic, they can cross that core. Then we have our transportation. Peptides are dissolved in the plasma. Why? They're water soluble. Steroids cannot be transported through my blood by themselves. They need those carrier proteins, albumin being one of them, globulins being others. The receptors for my peptide is membrane, versus steroid is going to be intracellular. Peptides alter the intracellular concentration of secondary messengers. We have a cascade, goes and turns proteins either on or off. Steroids bind directly to my DNA, the steroid hormone complex, our receptor complex, and then turns a gene, the transcription of a gene, on or off. Onset of peptides, very quick. By very quick, we mean it could be minutes, it could be seconds. Okay, steroids takes hours, takes days, okay, to see that full thing. Duration is going to be very, very short for peptides and long. Some can be permanent, which we talked about. Examples of peptide hormones are things like insulin, ADH. Steroids would be estrogen, progesterone, testosterone. Those are good common ones that we have. All right. This looks common. Is this surprising to anybody? This chart and how we filled it out? Nah, we've seen this before. Okay. Let's try a question. And this question, Shelby, if you can set a timer for 1 minute, because this is definitely a longer question. When I see this, I see a nice long question. I see nice long answer choices. This tells me on test day that this is going to be a reasoning question. It's going to take me a little bit longer to get there. My assess, plan, execute, and answer is so very important on a question like this. And absolutely, I agree with Roberto. Okay, Roberto says that we could triage this. Okay, this is going to be a longer question. This may not be the first one that I want to do on test day. So I agree 100% with that. Okay? All right, 1 minute. Good luck. About halfway through, I'll pull up our poll. Good luck. All right, if you have not yet put in your answer choice, please do so. We're split on this one, okay? We've got about 69% of us with the right answer. So let's go through and let's really think about our assess, plan, execute, and answer. So we have a radio-labeled hormone that is added to a culture of liver cells. After a 4-hour incubation, the cells were separated, and the radioactivity was found to be primarily located in the nucleus. Which of the following conclusions about the hormone is most consistent with this observation? What does it mean if it is primarily in the nucleus? It had to have been able to get into the nucleus. Yeah, in the nucleus, Esther, here is huge. It must be a steroid hormone. All right, so I'm going to pull the poll off the screen. I'm going to pull up some tools so we can cross out. So now we look at our answer choices, and I definitely see patterns. I see peptide, peptide, steroid, steroid. Which ones do I eliminate here? A and B, gone. So I'm going to put a nice little mark through A and B. Okay, just like I would on test day, I'm going to right-click on it, get rid of that answer choice. Now, Esther, another clue is that it was added to the liver. Not necessarily, okay? Any type of cell that you add it to. We could have taken cells from the liver, from the heart, from the kidney, maybe ovary cells, something like that. We could have taken any cells. And if we separate them, what we're looking for is where is that radioactivity? Okay? Does that radioactivity end up on the plasma membrane, or does that radioactivity end up in the nucleus? Yeah, and Crystal's got a point. Liver cells really do, a lot. They really do just about everything. We love our livers, okay? You could use a 4-hour incubation definitely as another clue, because remember, peptide hormones should be minutes or seconds, okay, whereas hours or days could be what we're looking for, okay, for our steroid hormones. So definitely after 4-hour incubation. But the biggest one in here is the fact that it's in the nucleus, okay? But we could definitely use that 4-hour incubation, because if it was a peptide hormone in 4 hours, it's going to be gone. We're really not going to see it. Yeah, it would be fast. It would be fast. So that's definitely another clue here. But the biggest one is this puppy is in the nucleus somehow. So the only permeable hormones can get into that nucleus. So this has to be steroid. Now, very, very common answer choices for the MCAT. Okay, 2 of one type, 2 of another, and then a comma. After that is the, well, why is it there? Okay, so the reason it's there is because steroids have that hydrophobic region. They can cross that hydrophobic core of my membrane and get into the cell. So let's look at our answer choices here. C says it's a steroid hormone because steroid hormones contain hydrophilic regions which allow it to cross. What is the one word that is wrong in there that makes it opposite? Yeah, hydrophilic. Huge, huge red light. Huge red light. If it's hydrophilic, what type of hormone is it? There you go. Perfect. It's peptide. So C's a goner. D says it's a steroid hormone because steroid hormones function as transcriptional activators by binding to DNA. Yes, it absolutely allows them to bind to the DNA. Okay, and I agree with Esther. Our DNA should be found in our nucleus. Absolutely. Excellent. All right, this one, we're going to go ahead and do this one together because I want you guys to tell me what I'm looking for. Which of the following hormones would require a second messenger to relay its signal? First of all, what types of hormones give us second messengers? Peptide. Excellent. What is the ending or beginning typically on a peptide hormone? In. It is insane how many peptide hormones begin or end in in. Okay? So we have hormones that can't cross, so we know it's peptide, and we know it's got to be IN. Well, gee, that's easy. And we all know insulin is absolutely a peptide hormone. We also know testosterone and estrogen are steroid hormones. But even if I had no clue what this last one was, how do I know that this is a steroid hormone? Yep, it ends in OL. It ends in OL. Yep, and Esther's got a good point. Remember, here we're looking for the odd man out. Okay, 3 of them are going to be steroid, 1 of them is going to be peptide. All right, this leads us to our final learning goal for today. And this is to identify negative and positive feedback loops. This is a way of controlling how these hormones work. And we already said the vast majority of what we see in our bodies are negative feedback loops. So in a negative feedback loop, we're going to inhibit those hormones or inhibit those earlier enzymes. This is the vast majority of what we're going to talk about. So let's talk about a common one. Okay, this is going to be our angiotensin, our renin-angiotensin system that works in our kidney. So we see low blood pressure in the glomerulus of the kidney, and this has a positive effect on angiotensinogen. Renin is going to make that angiotensin 1. Then ACE, the angiotensin converting enzyme, makes that angiotensin 2. If you've ever heard of ACE inhibitors for high blood pressure, that's the enzyme that they act on. Okay? Then I get angiotensin 2, which goes and stimulates the release of which steroid hormone? Which steroid hormone? Oh, well, oops, it popped up on the screen. I didn't mean to click it that many times. You guys already have the answer. All right, aldosterone. Aldosterone goes to the kidney, and you guys already said it acts in the distal convoluted tubule and the beginning of the collecting duct. It increases the amount of sodium reabsorbed. Water passively follows that. That increases my blood pressure. If I see the right blood pressure in the glomerulus, that goes back and that inhibits my angiotensinogen, which means this whole pathway turns off. All right, should you know this pathway for the kidney? Yes, you absolutely need to know this pathway for the kidney. We go through that in the kidney sessions. So make sure that you know aldosterone. All right, let's talk about our negative feedback loops a little bit more. The final result of that pathway inhibits the hormone or the enzyme. So the final result here was increasing that blood pressure. So when I see the right blood pressure at the glomerulus, the whole system shuts down. Okay? We can also have things like when you do your cellular metabolism, your metabolism sessions, we see a lot of negative feedback in there, where the end product will go back and will stop the enzyme from working. Okay, so think of it as the final result. Okay, because it could be a result of higher blood pressure, it could be the result of a byproduct. Okay, positive feedback. This is the final result of the pathway, stimulates the process to start anew. Now, let's talk about oxytocin. Okay, oxytocin is something that we need to know. And oxytocin is released, causing uterine contractions. Uterine contractions cause more oxytocin to be released, which causes harder uterine contractions. So this just is that positive feedback, and it just keeps going and going and going. Does anyone know what actually shuts this off? Why does this stop? Yeah, birth. So the expulsion of both the fetus as well as the placenta is what is going to shut this off. Okay? HCG. HCG is the human chorionic gonadotropin ester. That is what actually causes us to maintain the lining of the uterus so that the embryo can implant. Okay? That's actually an LH, luteinizing hormone. It looks like the luteinizing hormone, so it keeps that around so we have more progesterone. So that's early on. And HCG is actually what causes morning sickness. Okay, so that definitely has something to do with pregnancy, but it's much, much earlier in the pregnancy. Okay. All right, so for this one, it's actually the physical movement of the fetus or the baby at this point and the placenta out of the uterus, and that's what causes it. That's what causes it. Oxytocin can also help to start to stimulate prolactin so that we get that lactation as well. Okay, so it's actually the physical movement of removing things from the uterus that stops this. But we can see that takes a while. Okay, and that is a very, very specific end product. So positive feedback loops are much more difficult to control, which is why we don't see these that often in the human body, okay? Lactation actually happens after birth, so usually it's a little while afterwards that we get that lactation, the actual lactation that does that, okay? So that's getting a little bit more into it, Chris, than we need to know, but it's actually massaging of the uterus that will help to stop that bleeding, okay? All right, now I feel like we're going through our OBGYN rotations. All right, so as we look at this, these are our positive feedback loops. Let's look at what we talked about today. Types of signaling. Endocrine. Endocrine goes in the bloodstream throughout the whole body. We mentioned exocrine, which goes directly where it's needed by a duct or a valve, like the pancreas's exocrine function goes directly into the duodenum to help us with digestion. We have autocrine, which is going and helping us, okay, and that's a self-signaling. Paracrine. Paracrine is nearby signaling, right? What's the difference between paracrine and juxtacrine? Paracrine and juxtacrine. Juxtacrine, we have to be abutting, we have to be touching either physically or we can touch through some sort of bridge, okay? Yeah, so it's near versus touching. You guys got it. Now, we didn't really talk about tropic versus direct, but we'll talk about that a little bit more in endocrine too. So tropic means that it causes other hormones to be released. Direct means that it goes directly and acts on what it needs to act on. Let me give you a quick example of tropic versus direct. Does everyone remember the anterior pituitary and the nice mnemonic to remember what happens in the anterior pituitary, what's released? Flat peg. Nice. Flat, FSH, LH, ACTH, and TSH cause other hormones to be released. Those are our tropic hormones. Okay, FSH and LH work with estrogen and progesterone. ACTH is going to be adrenocorticotropic hormone, and that causes other hormones to be released. TSH causes the thyroid to release T3 and T4. Okay? Now, our direct hormones are PEG. Okay? Prolactin. That goes directly to the breast tissue, causes lactation. No other hormones are released. Okay? Endorphins. That goes to where we've really injured ourselves to inhibit pain. No other hormones are released. GH is the growth hormone that goes directly to bone, to muscle, to cause the increase or the growth of that. No other hormones are released. Okay? So flat is our tropic, PEG is our direct. We talked about 3 major types of hormones: steroid hormones, peptide hormones, and though less common, still important, are amino acid derived hormones, which is 1 or 2 amino acids. We talked about our negative feedback loops and our positive feedback loops. All right. You guys did a great job today. Hopefully, you're feeling a little bit better about the endocrine system, different types of signaling, different types of hormone, and now when we see them in passages, just by their names, we're going to be able to tell the difference between a peptide and a steroid hormone, even if we've never heard of those before. Just by looking at them, we're going to know what they look like, okay? Anterior pituitary. Yep, anterior pituitary. All right, so hopefully you guys are feeling better. Thank you so much for joining us, and we will see you in another section. Make sure you go to Endocrine System 2 to get even more information. Have a great day. We'll see you guys soon.